Hello, everyone. I'm Professor Tao Sang from Huaqiao University in China. First, let me thank Professor Ghulam Hossain for inviting me to give a seminar at this International Brain and Biomedicine Conference. I am so much honored to present, to share our work with so many brilliant scientists at this conference. First, please let me briefly introduce myself. I did my PhD in neurobiology at University College London in United Kingdom. After finish my PhD, I did my postdoctoral training at Harvard Medical School. At Harvard, I worked with Chuck Stiles and Chris Walsh, those two superb scientists. In 2005, I was lucky to get faculty position at Cornell University Biomedical College in New York, United States, and improved to associate professor. In 2017, I came back China and helped Huaqiao University to build up their first school or medical school and becoming the founding dean of the School of Medicine. Our lab has been working on the development of the central nervous system for many years, and in particularly the brain and the spinal cord. We are also very much interested in understanding the causes of human neurological diseases. Today, I want to share some of the work in human brain and spinal cord. And these diagrams shows the real brain images of human fetal brains at different stages. You can see gestational week 17 until 30. During this long process, you can see the morphology change and the growth of the brain, and the cell migrate, and the neurons form axonal connections. So brain is probably one of the most complex structures in human body. And we just look at the cell numbers, you can see there are over 100 billion neurons in the human brain. But no matter how complicated the human brain is, there can be two major cell types. One, we call them excitatory neurons. One, we call them inhibitory GABAergic interneurons. So if we look at the cerebral cortex, all the excitatory neurons, they are born from the region, we call it ventricular zone. They born from these progenitor cells. And how these progenitor cells proliferate and survive and differentiate is a major research interest to us. But if we look at the interneuron, they are also born from a structure called subpallium. And all those interneurons, they can be divided into three regions. For example, the medial ganglionic eminence, MGE, in the subpallium, or lateral, LGE, and CGE. All those interneurons, once they are born from these three regions, they will undergo a long tangential migration and reach their final sites in the cerebral cortex. So recent years, people pay very much attention to the interneuron development because we are more and more realize that the balance between interneuron and excitatory neurons in the cerebral cortex is extremely important because the unbalance between those two cell types could be a major reason of autism. This has been more and more recognized by scientists. If we look at the interneuron subtypes in the cerebral cortex, we can name them according to their morphology. For example, the basket cells, chandelier cells, they have beautiful morphology, they have many different cell types. But how these different cell types are specified in the human fetal brain, this is the question we are interested. In the past, it has been very difficult to address this question, but nowadays, based using the single-cell RNA sequencing approach, we are able to do it. Single-cell RNA sequencing, basically, you take a piece of brain tissues and dissociate those cells into single cells, and then you can use the 10x genomics platform and to sequence the RNA's transcription profiles in individual cells, and then they gather information of one cell, and then you know the overall of gene expression profile. To work on this project, two excellent students, Yuan Yu and Zhiwei Zheng, they worked on this project. What they did is that they collected the subpallium tissues from human and get their human fetal tissues from gestational week 9 to 12, and then use the single-cell RNA sequencing. And interestingly, you can say that those cells, they can be divided into several groups. That means there's a very high diversity of interneurons in human fetal brains. But no matter how complete they are, if we look at their gene expression profile and even look at their gene expression position using the in situ sequencing approach, you can see that they can be divided into two major regions. One is called the ventricular zone. You can see we use the pseudo color to label them in green. And one is in the subventricular zone. We use the purple color to label them. So this is very similar to the cerebral cortex. All those progenitor cells, they can be divided into ventricular zone and subventricular zone cells. And if we look at those interneurons derived from the MGE, you can see they can be grouped into 5 subtypes according to their combination of gene expression. And then those cells, they migrate tangentially into the cerebral cortex. So whether they are also specified, and when we look at their lineage development, we find one interesting feature is that they can be split into 2 lineages. We find that lineage 1 highly expressed gene like a MEIS2. But if we look at the gene expression position of MEIS2, they mostly located in the dorsal region of the MGE. On the other hand, the lineage 2 highly expressed gene like LHX8 and ZIG4, they are located in the ventral region of the MGE. So this suggests that the interneuron precursor cells, they are positioned at different regions in the MGE, probably determine their migration route later on. If we look at the interneuron precursors in the LGE, they can be divided into 11 subtypes, but they also can be grouped into 2 types. One is in the dorsal LGE, one is in the ventral LGE. To the LGE specific interneurons, and if they are in the dorsal, they mostly are involved in the visual system development. And if they are in the ventral, they are involved in the neuron projection development. So the third question we look at is that the diversity of interneuron precursors, how they are specified at later stage or early stage. On the left is the diagram of different cell types in the cerebral cortex. You can see they are located in the layer 1, layer 2, 3, and layer 5. But when we collect their gene expression profile with their adult marker, we find there's a very nice connection even at gestational 9. So this suggests that the diversity of interneuron neurons, they have a very early acquisition. That means their cell fate or cell types are predetermined even at fetal stage. So we published this work by end of the 2021 in Nature Neuroscience, and we have drawn 3 major conclusions. The first is that interneuron progenitors and precursors in human fetal subpallium are very high diversed. And the second is that we find the interneuron precursors in the MGE and LGE, they simultaneously acquire their temporal and spatial identity. They can be divided into dorsal and ventral subtypes. And the interneuron subtypes can be can be specified even at fetal stage. So this suggests that there's an early acquisition of cell fate for the various interneurons. So in the end, I want to share some unpublished work in the human spinal cord. And you can see human spinal cord, there's two major cell types. One is motor neuron and one is interneuron. So the human spinal cord neurons are very complicated. They have about 20 distinct cell types. So the question to us is also how they are specified. So a PhD student, Jing Wang, worked on this project. She collected human fetal spinal cord from gestational week 8 to 12 and used the RNA sequencing approach to analyze their cell diversity. And you can see again, it's very similar to the subpallium. They can be divided, into different cell types at very early stages. So, for example, they're just interneuron and motor neuron. They can be divided into 13 subtypes, and they have the progenitor for motor neurons and also progenitors for interneurons. But when we analyze those progenitors very carefully, we find one interesting group of cells. We call them common progenitors. They can be specified in 5 subtypes. Interestingly, we find that these common progenitors, they can give rise to motor neuron progenitor type 1, and then motor neuron progenitor type 1 will become type 2 and type 3, and later on mature into motor neurons. And these common progenitors also can give rise to interneuron progenitors, and then these interneuron progenitors will differentiate into various interneurons. So interneurons are very complicated in the human spinal cord. There are at least 11 interneuron subtypes aligned from the dorsal to the ventral spinal cord. So what about their gene expression features? Interestingly, when we look at interneurons, we find they can be grouped into 13 subtypes. And particularly, when you look at all those gene expression profiles, they correspond very well with their position in the spinal cord from the dorsal interneurons and to the ventral interneurons. So this suggests that even though the interneurons are very complicated in the human fetal spinal cord, they are already specified by combination of gene expression at very early stages. So to summarize my talk today, and we find that there are 2 types of neuroprogenitors. They reside in the ventricular zone and the subventricular zone. They are very similar in the cortex, subpallium, and spinal cord. So probably this is the common theme of central nervous system development in the human tissue. Second is that we find single-cell RNA sequencing can be a powerful tool to capture the diversity of neuronal subtypes in the human fetal brain and spinal cord. And third, we find that the cell fate determination can be a very early event. So it happened very early in the human fetal brain and predetermine their fate for the adult stage. So I'd like to thank all the people in my lab, and we have 2 lecturers, and the rest, they are all PhD students and master degree students. They work very hard to generate all those results I've presented today. And I also want to thank the funding support while I worked in the United States, for example, the National Institute of Health, and also when I come back to China, the National Natural Science Foundation of China. And in particular, I want to thank all my collaborators, Greg Hannon at Cold Spring Harbor, Tyler Jacks at MIT, Yunan Jian at UCSF, while I worked in America. And after I came back China, I was so fortunate to set up a very active collaboration with Professor Ghulam Hossain at Government College University in Pakistan. and Wenjie Cai from Fujian Medical University, and Zhiheng Xu from Chinese Academy of Sciences. And thank you again for attending my seminar. I'm so honored to share my work with everybody here. Thank you.